All right, BIM 100 students, let's get you ready for Professor Pasquinelli's final. In this quick power-up, we're going to dive deep into the key experiments from Lecture 7. Think of this as your guide to turning that homework into some serious exam points. Let's lock in these concepts so you can absolutely ace it. So, here's what we're tackling today. We'll start with a quick power-up, then jump into the hunt for new LNC RNAs, figure out how to track them with microarrays, and decode those results. After that, we'll cover how to identify an unknown mRNA, and we'll wrap it all up with some essential pro tips for the final exam. Okay, first up, final exam power-up. We're turning Homework 7 into exam points. Look, the goal here is not just to review the answers. It's all about digging into the logic behind them, because that is exactly what Professor Pasquinelli is going to test you on.
Right. So it's all about moving past just memorizing facts. You've got to understand the why behind every single experiment. Professor Pasquinelli's exams are famous for this. They really push you to think like a scientist. That means we're going to focus on the logic that connects the dots, from the initial question to the right technique, all the way to the final conclusion.
Section 2. The hunt for new LNCRNAs, or the scientific quest after heat shock. Okay, let's jump right into the big scenario from your homework. Imagine this. You've just blasted some cells with heat shock. Now what? The big scientific question is, what new RNAs are these cells making in response to all that stress?
So this is the big challenge, right? You've heat-shocked your HeLa cells. How in the world do you find brand new, long non-coding RNAs that might be popping up? I mean, you can't look for something if you don't even know it exists yet. So what you need is a technique that's like casting a giant fishing net, one that can catch everything that's being transcribed. And that brings us to our first key tool. And that tool is RNA-seq. You've got to know this one. It's your absolute go-to for discovery. Why? Because it's completely unbiased. You don't need any prior knowledge of what you're looking for. You literally just grab all the RNA from your sample at that specific moment, turn it into cDNA to make it more stable, and then you sequence everything. It's like turning on the lights in a dark room. Suddenly you can see all these new transcripts you never knew were there. So how does this actually work? It's a pretty logical four-step process. First, you have to isolate your RNA from both your control cells, the ones just chillin', and your experimental cells, the ones you heat-shocked. Then you make your cDNA, chop it up, and stick on these little sequencing linkers. Step 3 is all computer work. You map all those millions of sequence reads back to the human genome. And here's the payoff, Step 4. You start looking for RNAs that are only in your heat-shocked sample. And the final check? To make sure it's a non-coding RNA, you confirm it doesn't have a long open reading frame. You know, that's the signal that screams, make a protein here. Next up, Section 3, the time course, using microarrays. We're going to measure the expression of our 5 new candidates. Okay, so RNA-seq was a huge success. We found 5 brand new LNC RNA candidates that seemed to be turned on by heat shock. Awesome. But science never stops, right? Now we have a follow-up question. How does their expression change over time? For this job, we need a totally different tool. And this is where the microarray just shines. Here's the key difference you absolutely need to remember. If RNA-seq is for discovery, finding new stuff, a microarray is for quantification, when you already know what you're looking for. See, we now have the sequences for our 5 candidates, so we can design custom DNA probes for each one, stick them onto a glass slide in tiny little spots, and then see how much of our cellular RNA actually sticks to each of those spots. Okay, exam alert. This is a huge pro tip, so listen up. Whenever you get an experimental design question on the final, do not just start listing steps. The very first thing you should do is state your control condition. From microarray, that means you have to be super clear about your baseline. For instance, you'd say something like, my control is cDNA from the non-heat shock cells, which I'll label with a green fluorescent dye. My experimental sample is cDNA from the heat shock cells, which I'll label with a red dye. That clarity is going to get you points. All right, Section 4, decoding microarray results. Let's talk about what those colors actually mean. The cool thing about a microarray is that it gives you this really powerful visual snapshot of gene expression, but it only works if you know how to read the language of the colors. And the logic is actually pretty straightforward. Remember how we labeled our heat shock sample red and our control green? Well, if a spot glows bright red, it means a ton of the red-labeled cDNA stuck to it. Conclusion? That lens RNA is upregulated by heat shock. If the spot is green, the opposite is true. It's downregulated. Yellow is the middle ground. It means you have roughly equal amounts of red and green binding, so no change in expression. And if a spot is just black, well, that means you got nothing from either sample. So the key takeaway here is that it's all about relative amounts. The color and how bright it is tells you the story of a competition. It's like a tiny tug-of-war on each spot between the cDNA from your control sample and your experimental sample. The final color just shows you who won. It's a beautiful, visual way to see the ratio of expression.
Okay, let's switch gears a bit. Section 5, Identifying an Unknown mRNA Using Bioinformatics. This was the other big task on Homework 7. So now, we're not trying to discover something brand new. Instead, it's more like a mystery. We've been handed a short, unknown RNA sequence, and our job is to play detective and figure out exactly what it is. Now, the homework answer key told you the answer. The sequence was a 100% perfect match to MIR122, a known human microRNA. But just knowing the answer isn't enough for the final. The real question, the one you'll get tested on, is how would you figure that out on your own? And the answer is bioinformatics. The standard way to do this is to use an online tool called BLAST. You literally copy and paste your mystery sequence into BLAST. But here's the crucial part. You don't just search it against everything. You tell it to search specifically against a database of known human microRNAs. That way, you're asking a very targeted question, and you can see if you get a significant match. Now, here's a detail that will really make your answer stand out. You'll see something called an e-value, or expect value. Think of it like this. It's the number of times you'd expect to find a match just by pure random chance. So, a tiny e-value is what you want. It means your match is significant and not just a fluke. And as the homework pointed out, even with a 100% match, the e-value isn't 0 for a short sequence like an mRNA. Why? Because it's so short, there's always a tiny statistical possibility it could match by chance. But a super small e-value tells you, nope, this is the real deal. Okay, last section. Let's wrap this up with some final exam pro tips. This is all about how to get inside Professor Pasquinelli's head and think like a scientist. We've gone over the experiments, now let's talk strategy. This is your checklist for success on the final. Number 1, always state your controls. First thing. Number 2, explain the why. Don't just give a recipe. Explain why you're adding the linkers or using a particular dye. 3, connect the techniques. Understand the story of how RNA-seq for discovery leads you to using a microarray for quantification. 4, and this is a big one. Interpret your data, don't just describe it. Saying the spot is red isn't enough. You have to say the spot is red, which means this gene is upregulated by heat shock. And finally, please read the question carefully. Little details like which sample got the red dye can change the whole answer. So I'm going to leave you with this final thought experiment to chew on. This is exactly the kind of synthesis question you could see. If you had data from a DNase footprinting assay and a microarray, how could you put those two pieces of the puzzle together to prove that a specific transcription factor is flipping the switch on a whole set of heat shock genes? Thinking about how to weave together data from different experiments, that is peak scientific thinking, and it's the best way to prepare. All right, you are officially powered up on Lecture 7. Take this way of thinking, apply it to the whole course, and you are going to do great. Good luck on the final.